Today we'll talk about generics and biosimilars in a different context than intellectual property. Usually, generics and biosimilars focus on copy of somebody else's work. This presentation is designed to move you, the listener, to activism, to be proactive about what the future ought to hold. So, are you a branded, a sponsor, a generic, or a biosimilar company? Do you represent such companies? What do you do about a situation, about the situations for all of these companies, branded or generic, biosimilar or sponsor, where your client, your company, your research involves copying what somebody else has done? Now, copy is an important factor in all of these situations. It doesn't matter whether you are a sponsor, an originator, or someone in the biosimilars field. Copying is something that you do. So here's a roadmap of what we will talk about today. Why did you choose this path? And who likes this path? Do you like it? Does your CEO like it? Do your researchers like it? What is a copy? What expertise have your scientists gained? And what are the risks and benefits? And what those risks and benefits might lead to in terms of SuperGenerics and BioBetters. So let's start with this discussion about the benefits. What's the benefit of copying? Of course, it's great for consumers. We copy other people's work because it allows us to lower the cost. The government wins in this situation. Insurance companies win in this situation. Government insurance wins in this situation. Consumers win in this situation. In addition, for the company or your client or your research effort, there's little or no risk. As many of you know, when you are conducting research in the pharmaceutical or biotech area, out of the 10,000 or 100,000 compounds or biologicals that you may develop in the laboratory, only 1 or 2 of them ever make it through a phase 2 FDA trial. And out of the 100 or so that make it through the FDA trial phase 2, only 1 will make it through phase 3. It costs a huge amount of money to go through all those clinical research efforts to do the clinicals through phase 3. Copying of something that is already approved by the FDA, or in Europe, for example, by the European Regulatory Agency, excuse me, involves little or no risk or expense in terms of clinical research. It's almost a guaranteed regulatory approval, so it reduces your development costs for copying. So what did you do to copy? Well, you spent years looking through the literature, looking through the orange book, looking through the purple sheets, looking through the European Regulatory Agency documents to identify small molecules, biologicals, other items that you think would be worthwhile and profitable to copy. When you set about looking at these things in the laboratory, you looked at their physical features, their chemical features, their biological features. You tested them in some animals, and you tried to copy the process. Now, for pharmaceuticals, it's very easy to copy the process. Pharmaceutical processing, pharmaceutical synthesis of small molecules is a fairly straightforward process, a fairly straightforward technique, and there aren't any guesses involved. But when it comes to biologicals, of course, there's a big difference. In any event, your duplication came very close to duplicating the impurities and the side products and everything else that was in the original product, so that you could have that almost virtual guarantee of FDA approval. So let's look again at copying. Why did you copy? You know, sometimes we say that copying is wrong. It's like plagiarism in school. Do you remember in high school or even in junior high when one of your fellow classmates was found to have copied somebody else's work or copied something out of a brother or sister's notebook, or even worse, copied a poem from Shakespeare and handed it in as an original. And of course, in that situation, the teacher would accuse you, or accuse your colleagues, accuse your fellow students of copying, and it was a bad word. It was a pejorative word. You have copied. That's wrong. You need to do it originally. Well, in pharmaceuticals and biologicals, of course, copying is legal. It's the Hatch-Waxman Act and the BPCIA, the Bixia Act. It's the two acts that legitimize copying of regulatorily approved pharmaceuticals and regulatorily approved biologicals within certain constraints. So what are those constraints? There's minimal or no risk of regulatory agency rejection, 110%. It's a sure thing for safety and efficacy. What do we mean by safety and efficacy? Well, safety and efficacy are the two buzzwords for obtaining approval by the FDA in the United States or the European Regulatory Authority in Europe. You have to demonstrate that your product, or pharmaceutical or biological, is safe. Number one, it doesn't kill people and it doesn't cause bad side effects. And number two, it's efficacious. It does what you say it will do. It provides the benefit, the treatment of the disease that you say it will accomplish. So copying affords you, the copier, a lower clinical cost, a much lower clinical cost, fewer preclinical proofs, but you still need to show the same chemical and physical parameters. And that is a very easy factor to take care of. So why do you copy? Number one, it's legal. Number two, it's an easy route to earn big bucks. So successful copying, yes, you have won the race. What do you do now that you have success? Will you make additional generics and biosimilars? Will you continue copying? It's certainly an easy path to follow. No clinical research is involved, or very little clinical research is involved, and it's profitable. At least you think it's profitable. So the big risk of copying, however, is that it nevertheless takes years to develop. Once you show that something can be copied, that a biological or pharmaceutical can be copied, you generate competition. There are other companies out there who will do the same thing. They will see, oh, this product, this pharmaceutical, this Lipitor, for example, can be copied, and we don't have to worry about patenting situations anymore. So we can copy it too. And so all of a sudden you have generated competition. The competition is brutal. In fact, in generics, it's often a race to the lowest cost product. So a price squeeze is what happens. And let's take an example of Pfizer's Lipitor, atorvastatin. It was developed in the late 80s by Warner Lambert and Park Davis. Pfizer marketed Lipitor, and later it bought Warner Lambert for huge billions of dollars. It wasn't the first statin on the market for control of cholesterol. The first one was Merck's lovastatin and simvastatin. However, Lipitor was the most successful statin on the market. And between 96 and 2012, Pfizer made, this isn't the gross, this is the net. Pfizer netted more than $125 billion worldwide from selling Lipitor. So the extension patents, however, expired in 2012 for Lipitor. And all of a sudden, two companies jumped into the generics business, Watson and Ranbaxy, and now they're producing atorvastatin. Of course, they can't use the same brand name, the same trademark, Lipitor, as Pfizer had. But now that same compound, atorvastatin, is sold as Statter, it's sold by Teva as atorvastatin. It's sold as these other kinds of trademarks, and actually Pfizer's generic arm sells it as Zerator. All of these products are now on the market. Here's what happened to the price. The price of Lipitor per pill was a little more than $200 per month. Now I take Lipitor, and I've taken it for years, and I remember when all the other statins were coming off patent about 3 years before Lipitor came off patent. There was a big problem with my insurance company. They kept telling me I need to move to simvastatin, one that is not on patent anymore. And my MD put me on the simvastatin, but it didn't work as well. So he moved me back to Lipitor, and I had to pay the difference in price. But now the price for Lipitor is less than $10 a month. In other words, it's about a 22 times reduction, or it's 31 cents per pill instead of $6.75 per pill. So what's the bottom line? As Carl for Choice Hotels says, low price guaranteed. And you can count on the fact that this is what happens in the competition market for generics. What about biosimilars? What about biosimilars? Remember the FTC, not FTV, my apologies, what the Federal Trade Commission prediction in 2013 was about biosimilars. Because biosimilars are so complicated, they're huge protein molecules, and it's going to be very difficult to manufacture them. They're not like generics. They don't involve a simple processing synthesis. They involve a very difficult fermentation and recovery and purification process. So biologicals in the biosimilar situation will resemble brand to brand pharma competition rather than brand to generic competition. And the FTC said that these predictions will lead to better profitability and everybody will benefit, except you consumers, of course. But is that right? If you copy and sell a biological, will you earn big bucks? Well, I am here to tell you the cost drivers for biologicals. Will the cost become lower? You bet they will. Here's an example. Univercells production, the single-use high-cell density bioreactor. It's not a huge fermentation tank anymore, no filtration equipment, no chromatography, no huge chromatography columns, no large pumps. It's a very simple apparatus. It produces, purifies, and dumps out the product in a very simple one-room machine. And the cost involved is very similar. So do we need to worry about these production costs, or will clinicals still be the big killer? So if you choose the big killer, like etanercept, like Herceptin, like Humira, and you have to run through the clinical costs for these sellers, would that be a barrier, even though the production costs are lower? Actually, no. Here is that typical example of the difference between the originator development and the clinical costs. Clinical costs are huge. And for biosimilars development, the world is flipped upside down. And here's a real example of TNF alpha production without the processing improvements. And so you can see by this chart that for the clinical developments from phase 1 to phase 3, the costs dropped by approximately 2/3 to 1/2, going from 2007 to 2016, relative to the originator's clinical costs. So clinical costs plus the production costs will push these down. Here's an example of what I mean with biosimilars. Etanercept. Etanercept is a TNF alpha inhibitor. And the patent was set to expire in 2012, and the price in the United States was $2,225 per month. This is a drug that's given for rheumatoid arthritis, and it's patented by Amgen. The U.S. patent will expire in 2028. What will happen when the patent expires? Well, in Europe and in other places, the etanercept patent has already expired, and there are a number of producers, 24 producers of the biosimilar of etanercept. And what will happen? An example is what has happened in Switzerland. The price has dropped by 1/2 already, from $2,000 to approximately $1,000 per month. Yes, that's still a lot of money compared with 31 cents a pill, but let me tell you, paying half of the fee for this biological when the benefit is so great is a wonder. So what are the risks and the future for biosimilars? Well, I can tell you that the risk for biosimilars is going to be just like the risks for generic pharmaceuticals. There's going to be competition. It's going to be completely price driven, and the lowest production cost will win the day. It's a school of sharks. That's where you'll be swimming. So what is your fate? Success or failure, and what does it mean? Well, first of all, there's timing. Generics typically take 3 to 4 years to develop. Biosimilars are a little short of 8 years. That's a long time for developing a product. And what is the alternative to this? There's a better alternative. You need to think 3 steps ahead. You need to develop some exclusivity, gather a competitive advantage, and get your own patents. Now, in the super generic, or rather in the generics and the biosimilars field, saying you should get your own patents may seem like blasphemy. Of course, the idea of generics and biosimilars is we don't want to deal with patents. The products ought to be free for use so that we can compete on a price basis. However, as you have just seen, what happens is you have multiple competitors, and although it's good for the government and it's good for the consumer, for you, the producer, for you, the patent attorney who's counseling the producer, it drives the price of the product down. It means that the company is making less profit. The company needs to reduce its research efforts. The company needs to cut down on development of new products. It's a very difficult situation to be in. Is there a future? Well, what's the hidden benefit of all this? Let's think about this. What is the hidden benefit of having developed a generic or a biosimilar? Well, wait a minute. You need to do clinical development no matter what. And you have reams of clinical data. What does this mean? Well, let's look at your researchers. Your researchers worked for years, 3 or 4 years for generics, 6 to 8 years for biosimilars, on developing an understanding of the technology for producing this kind of product. They have expertise. They know how to make this kind of situation work. What is that expertise? Well, wait a minute. In generics, what your researchers have done is they have tried to duplicate, and they've tried to duplicate something that happened 10 to 14 years ago. Your biological researchers are duplicating, I emphasize duplicating, something that happened 15 to 20 years ago. Think about what's happened in the biologicals and the biotechnology field in the last 20 years. It's like we have invented a faster than light rocket ship, and we are now heading for the Andromeda galaxy relative to how it was 20 years ago. Your researchers know today's cutting edge research. They're smart. They're not 20 years behind in the knowledge. You need to turn them loose. And they also have the practical experience for working on these generics and biosimilars. And they know how to make your product better, cheaper, and more effective, and in fact, make more money. How do you do that? Well, you switch to SuperGenerics and BioBetters. And what do I mean by SuperGenerics and BioBetters? Well, let me tell you. There are a number of things you can do. You can make a structural variation. You can make a different pharmaceutical formulation. And let me give you an example. Consider the supergeneric Eligard. Eligard is an injectable single body liquid that when injected forms an implant in the body. And it's the chemical castration drug. The nonapeptide, known as leuprolide acetate. There was a competing product on the market. It was already approved by the FDA. Its name was Lupron Depot. It was produced as preformed microparticles in a bioabsorbable polymer, and it was injected as such into the muscle. Problem was that Eligard was the same kind of drug. It was the same kind of delivery system, a sustained release polymer, but the Eligard was not multiple microparticles. It was an in situ implant, so it was very easy to administer. If the implant was a bioabsorbable polymer with a leuprolide in it. When you injected it, it converted from a liquid to a solid. Oops. And so this is an example of a situation where you have a super generic. What do I mean by super generic in the Eligard situation? Well, a single injection of Eligard lasts at least 6 months, whereas the Lupron Depot lasts a maximum of 3 to 4 months. It created huge patient acceptance. Why? Because the injection is painful and it requires a clinical visit. It's painful because you get shot in the abdomen with this material and it hurts. Let me tell you, it hurts. And so, which would you choose? To be stuck twice a year with a big bad needle, or 4 times a year at least with a big bad needle? So here is an example of something that was really a change of the formulation, but the elements are the same, the polymer, the drug, and as a result, it became a big seller, and Eligard was patented. It provided exclusivity. The strategy here is to file patents at your early date, file before the technical or clinical or biological data are published, and consider invention by selection and all the other fancy tricks that patent attorneys have in their portfolios. And you need to consider the evergreening possibilities. And we can talk about these at length at another seminar, but just be aware that once you get into the area of super generics and super biosimilars, you have the ability to obtain patents and exclusivity and garner a higher profit. So what about IPR? You say, wait a minute, IPR is the big boogeyman for pharmaceutical companies, and of course, an IPR earthquake has already happened. No longer is the broadest reasonable interpretation of claim terms the way that the patent court at the patent office looks at claims that are under IPR investigation. Instead, it's now the same Phillips interpretation used by the federal district courts. So it will make life a lot easier for patent holders. And let me tell you, it'll also make life a lot more difficult for generics and biosimilar companies that want to challenge patents in an IPR process. And of course, we all know what the proposals have been by Hatch, at least, to prevent using IPRs in situations where you have a Hatch-Waxman possibility, but it doesn't look like this will happen. In any case, the point is that IPRs are not the big bugaboo that we thought that they were, and especially for pharmaceuticals and biologicals, the success rate is a lot higher in IPRs than it is for patentees than it is for the computer and software situations. So super generics and BioBetters, that will allow you to avoid being in the shark school. The expense of production and clinical are becoming precipitously lower for generics and biosimilars. It's a race to the bottom for price. If you think about allowing your researchers to use their current knowledge and their experience in developing those generics and those biosimilars, turn them loose to develop BioBetters and SuperGenerics. These don't have to be E equals MC squared. They don't have to be a leap forward. They can be a minor change, just like Eligard was. And of course, the Eligard folks will say, no, it was a major change. But Eligard was a change. It provided a patenting technology, and it was a patent that withstood challenge. And so those kind of changes will afford you a better way of developing a product that will deliver to the customer, to the patient, better properties, and it'll deliver to you and your clients and your company a better patent position. So that is the end of my somewhat different talk about IP in the terms of practical aspects, and to try to move everybody in the business of generics and biosimilars to think about being proactive and moving the world forward to help the population at large with better products, not products that were developed 20 years ago, but products that are being developed today. Because after all, in 20 years of research in biologicals and biotechnology, look at how far we've come. Don't you think that these new products or I should say, biologicals that were developed 20 years ago could be made better by using that new technology, by using ways to administer them to increase their lifetime, and on and on and on. You know the ideas, you know the techniques, and so what I say is get on with it, move up, and think about being proactive. Thank you.